So open this talk about biophysical fiction versus reality, I'm going to start with a science fiction book. This book, Jules Verne, you may be familiar with. He's a famous 19th century science fiction author, wrote books like 20,000 Leagues Under the Sea. This is the last book. Can you hear me? Sorry, I'll be closer to it. Jules Verne is a famous 19th century science fiction author. He wrote this book called Paris in the 20th Century in 1860, but his editor said to him, "My dear Verne, even if you were a prophet, no one today would believe this prophecy. They simply would not be interested in it." And so he shelved this book, and it was only published over 100 years after his death in 1994. But what's interesting about this is he envisioned a Paris in 1960 that had gas-powered cars, it had electronic music, neither of which would have been available in 1860. He envisioned a centralized global financial system. He also envisioned some version of the internet in this time. Now, it took us about 30 years longer than Jules Verne predicted to realize the internet, but it shows how things that seem fantastical or unbelievable at one time can become reality. Now, I want to give everyone a disclaimer up front because a lot of the things I'm going to talk about have an ethical dimension to them. I will not be able to get into full detail of the ethical issues in play, but I'll try to give at least a highlight or flag things that are unethical. And you guys probably heard some laughs. This picture is Jeff Goldblum playing Dr. Ian Malcolm in the adaptation of the novel Jurassic Park. And he has a quote in this scene where he says, "Your scientists were so preoccupied with whether they could, they forgot to ask whether they should." So I will try to highlight these in certain areas along the way. So with that, I'm going to turn to a first example of a surprising biophysical technology, although perhaps not anymore, is genetically modified organisms. And to do this, I just want to recap that the instructions for building us is in our DNA. And there's a biochemical process that's a little bit, that we can make an analogy to our computer with. The DNA you could think of like your hard drive. The mRNA is a little bit like RAM. And then the proteins might be the libraries or the computer code that actually executes what you want, maybe the CPU. In our bodies, at the level of single cell, the proteins coordinate metabolism, and metabolism works together with the cells to give the function of the organism, roughly speaking. So this all goes to say that if the DNA in our bodies can be altered, we can alter major phenotypic things about us, major things that show up in our bodies, like our hair color, our eye color, your susceptibility to disease, and so forth. Now, you might, if we think about it, we have been modifying organisms genetically since we've domesticated animals like the dog or the cow. But this really increased a lot after the discovery of evolution or the observance of evolution by Charles Darwin. So in the Victorian era in England, the Kennel Club was established and people really started to breed dogs for specific physical characteristics. So there's also, I think many times we have used cloning or chimeras in agriculture. I'll get to chimeras a little bit later, but bananas are grown through a self-cloning process where you can take part of the banana and reestablish the entire tree from what you had before. And then to reproduce, this ensures that the desired you get a consistent fruit each time from each tree. However, this has the downside of making you very susceptible to things like funguses. And this is why you have bananas that are threatened, because the fungus, they have no natural resistance to the fungus, and it can spread and wipe out all of the bananas because they all have the same genotype. And so this is an example of genetic manipulation can have unintended consequences. Now, before 1978, but in 1978, we had the first example of genetically modified organisms being modified at the molecular level to do industrial processes. And so if anyone has used insulin, before 1978, we had to extract it from animals, which was really difficult and expensive. And since 1978, you could use microorganisms to make the insulin for us, which made it much cheaper. In fact, even today, now they can have a technique to make it through plants, which hopefully would make it even cheaper than it currently is. Now, just a few years later, there's this example of making this tobacco plant glow. And basically, they took a gene from fireflies, they put it into the plant, and then if you water it with the right protein mix, it will light up. And so this was in Science in 1986. If you are from Canada, you may have eaten a genetically modified animal where this Aqua Advantage salmon, which was developed in 1989, but only more recently gaining approval in Canada. In fact, it went through so many regulatory hurdles in the United States that I believe the company trying to make it collapsed. The idea here was that they took a gene from the Pacific salmon that grow a little larger, and they spliced it into the Atlantic salmon. And what it enabled the salmon to do is they would grow year round. So you can see an unmodified salmon in the foreground and a larger modified one in the background. But of course, people are very concerned about what happens if this gets into the animal populations, because in theory, the large salmon are not supposed to be able to reproduce. But another Dr. Ian Malcolm quote, "Life will find a way," is always a concern. And if formerly Monsanto, now Bayer, made a number of crops that were genetically modified to resist the pesticide, herbicide Roundup. And this also this could in principle boost crop yields, but it also led to lots of questions around intellectual property because they wanted to make sure that the farmers had to buy the seed from them and no one else. It also led to people using perhaps more pesticides than they would have otherwise because their plants were resistant to it. And as an example of an unintended consequence, some of the weeds that were initially sensitive to Roundup became resistant to it. And so now Roundup doesn't work. They're trying to use other pesticides like dicamba, which was just people didn't want to use it because of its side effects. And so you have this problem of managing these crops once you put them into the market. All right, so we talked a little bit about, broadly speaking, genetically modified organisms, but I want to talk about one specific technology that's come into vogue in the last 10 years. Yeah, I heard someone say it, CRISPR, because it really has supplanted a lot of the other technologies that were used for these older examples. So this starts from this timeline where we have our first person to originate this research was Francisco Mojica, who was studying bacteria that grew in very salty water. And he wanted to understand why some bacteria grew better in salty water than others. And to do this, he decided, I'm going to look at their DNA, I'm going to find out what they're doing. And when he did that, he realized that there were these repeating sequences and these repeating patterns, but he could not figure out what they were. These patterns were what we call palindromes and like a palindrome, like the word race car is spelled the same forward and backwards. You can also make palindromes in the DNA sequence. And these are usually important because they often signal special sites where proteins can cut the DNA. He, in 2005, was putting some of these sequences into the newly developed gene databases. And for a long time, nothing would come up until finally one day he put one of these sequences in and it came up as resistance to viruses. And so from that, he was able to conclude that these sequences had something to do with an adaptive immune response inside bacteria. By adaptive immune system, we have our own adaptive immune system in our memory B and memory T cells. So if you get sick from a virus, hope your body is primed to respond to it. And so this system was priming bacteria to respond to viral infections. Now, Emmanuel Charpentier discovered another part of this system, a protein called Cas9. She discovered that the bacteria uses this Cas9 as part of this immune system to cut out pieces of DNA. And she found that you could actually direct this protein to go to specific spots in the genome. Before this, we knew about restriction enzymes. These are proteins that cut DNA at specific sequences. But if you didn't have the right sequence, it wouldn't cut the DNA. Now, you could cause DNA to be cut anywhere in the genome. And Virginijus Shinxis cloned Cas9 into E. coli, so we showed that not only was this something that existed in bacteria in the wild, but you could actually transport it between organisms. So this is a step that allowed it to become more of a technology that you could use at will. This is all now in 2011, and he also showed how Cas9 works biochemically. So he did a lot of work on understanding how the structure of the protein related to its function. And then Jennifer Doudna, who you may have heard of, with Charpentier, showed that you could use just one single guide RNA inside the CRISPR to direct it anywhere you want in the genome. And in 2013, Feng Zhang showed that you could use CRISPR in eukaryotic cells. Those are like mammals, like our own bodies. So I have Emmanuel Charpentier and Jennifer Doudna starred because they won the Nobel Prize, but there was definitely some controversy because of how many other people contributed, and also who had the first ideas who managed to publish their ideas first versus having the ideas first. Now, I want to talk a little bit about the biochemical details of CRISPR-Cas9, just to give you an idea of what's going on inside the cell. As I mentioned before, bacteria and viruses have engaged in a war, if you will, against each other for billions of years. And this CRISPR stands for clustered, regularly interspaced palindromic repeats. So these are sequences of DNA that look kind of like this diagram on the bottom, where you have R, which is the repeating palindromic sequence, and then you have these S blocks. The S blocks are snippets of a viral genome. That's why you see the phage, the virus, diagrammed below them. Viruses do not infect the cells that have little pieces of their genome already in there. And that's because Cas proteins are acting like an immune system. They're sitting around in the cytosol, they're holding on to those pieces of DNA, and those pieces of DNA can be released when the virus infects to interfere with the viral genome. The virus needs to hijack, in order to reproduce, the virus has to hijack the bacterial cell. But if their DNA gets cleared before it can start hijacking the cell, it can't infect it. And that's what the Cas9 proteins help to do. Now, how does this become a technology? Well, if DNA is the software, if we can edit anywhere in the genome, then we can write our program or rewrite our own programming. And if Cas9 can cut the DNA, then we can do this anywhere that we want. So if we have this guide RNA, which is in blue, it's inside the Cas protein, which is the big blue blob. Cas protein can be targeted to any location in the genome, basically by designing your RNA to have the right sequence. So if you know what sequence in the genome that you want, you can make the RNA that's complementary to that sequence. And then Cas9 will go in, it will cut at that location, and you can insert the new DNA there. And Cas9 can be genetically modified, not only to rewrite sequences, it can silence production, it can edit sequences, it can also be genetically modified to recruit transcription factors and activate certain sequences of transcription. But I'll note that there are several limitations to our current, to the CRISPR as it was originally discovered, in that usually you can only target one sequence at a time. And there's been a lot of work in trying to limit off-target effects. And finally, if you are doing this, for example, in bacteria, the bacteria might not, you need to make sure that they hold on to the CRISPR that you're trying to put into their cells, and they might try to get rid of it, get rid of the biochemical system you insert. Now that I've talked about gene editing, I want to move up in scale to organisms and talk about cloning. And the key technique here is called somatic cell nuclear transfer. And I'm going to illustrate it in two videos. There's basically two steps. One is you need to recover the nucleus of the cell that you want to clone, and then you have to put that nucleus into a second cell in order, usually like an egg cell, to clone it. So here on the right-hand side, I'm going to show this video. You're going to see a pipette, and it's going to be stuck into the cell, and you can kind of see the nuclei. I believe there are two nuclei in here. And then the rest of this cell is just the cytoplasm. So as we watch this, you can punch the pipette in there, and then using the pressure of the pipette, you can actually suck the nuclei out of the cell very carefully. And you're going to see that it's the cell kind of has the proteins that are sticking to it until finally you can pull that protein free. And then you can also take a donor cell here where the nuclei have been removed and put a different nucleus or the desired nucleus back in the cell here. So this would be like an egg cell. And so you can push out the nuclei back into the cell. Now, while this is kind of a modern version of this technique, it's actually been around since the 1950s, when researchers did this in tadpoles of the African clawed frog. But it really gained a lot of prominence when Dolly the sheep was cloned in 1996. And what's interesting about, other than the fact that it's a mammal that was a genetic clone of a parent, it's also remarkable that this happened almost by accident, because the researchers were in the process of refining their techniques. And so they took an adult cell from one of the sheep, and they had done the direct nuclear transfer, and they were growing the egg in the petri dish, and they saw, hey, this thing is growing. It looks like it's developing like a normal embryo. Well, let's try to progress to the next stage of our experiment, where we're going to try to implant the embryo in a donor calf, these are steps that are very similar to in vitro fertilization that is quite, now today quite commonplace. So it was very surprising that this adult cell could start to behave like an embryo. And the success of this experiment directly inspired the researchers who won the Nobel Prize for reprogramming adult cells to behave like stem cells years later. And it's also inspired work here at the center by myself and Adelson to understand how turning on and off genes can change cell behavior. So this is where a computational project where we're trying to understand how gene activation and G activation relates to cell behavior, but with the hope is that one day this knowledge could be integrated into technologies that can cure diseases. Now, there's some fun stories about cloning, and if you listen religiously to This American Life some years ago, you might have heard the story of Second Chance. And so there was a Brahmin bull that was very docile, and he sadly died. His name was Chance, and his family heard about cloning, and so he started bugging researchers at Texas A&M. Hey, can you clone my bull? And we're looking at, they discussed it and they're like, well, we do want to test how old of an animal can we clone. So they agreed, and they did in fact clone the bull. And he looked a lot like Chance, and his owners thought he behaved in a very similar manner, being very docile. Now, if you've ever watched a rodeo, you know how powerful and dangerous bulls can be. And unfortunately, this bull did not maintain the same docile disposition and attacked his owner at least twice. And if you can watch the This American Life actually has video interviews with the owner, and you can see him sitting in the hospital after the bull has ripped his after he underwent surgery because the bull ripped his scrotum. Yeah, and he's saying, I still think he's going to calm down when he gets a little bit older. And it's funny and it's also heartbreaking because you can see him trying to cope with the loss of his bull and not wanting to realize what's happening. Now, perhaps a less funny story is about Hwang Woo-suk, who became a national hero in Korea because for his work on embryonic stem cells. But in 2006, he was disgraced because they found that he was fabricating his data. And even worse, he was using eggs gathered from his graduate students. I don't think I need to say how wildly unethical this is. But his subsequent career in cloning pets and dogs and working for countries like Libya, I guess, suggests that if you have biotechnical expertise and a total lack of ethics, you can still make a living for yourself. Um, that said, I want to point out, just take bookmark that, scientists rely on cultivating trust with the public. And so beyond his own work, it also destroys that trust. And that means that if we don't have that trust, that means we can't it makes it harder to bring technologies that could potentially save lives out and to the market. Now, I'll also want to mention the possibility of human cloning, which you might be wondering about. And so when identical twins form, you could think of this as like a natural cloning event. And essentially what happens is after the egg is fertilized, if it splits at an early enough stage of development, two individuals form. And this sort of embryo splitting has been used in animals because sometimes you want genetically identical animals, for example, if you are maybe mice for an experiment or potentially in agricultural applications. There's limited investigation of this in humans, but I must emphasize, it's also heavily regulated. Basically, the ethical consideration in humans when it's IVF is if a woman wants to have a baby, but is having a very hard time producing eggs on the stimulation that you go through, which involves injecting yourself with drugs almost every day over the course of a month and dealing with the side effects of putting, large amounts of hormones in your body. It's not something and then you have to go through surgery to retrieve the eggs for the IVF. If that is not to mention the expense of that, if you get a high quality embryo, sometimes there's an ethical question of should we split the embryo to give her another chance so that if this embryo, because not all embryos are successful, to avoid having to go through this again. Today, I think the technology is much with the rest of IVF is so much better that the chances of you being successful with it are much higher. But it raises this ethical question of which cost is greater, because obviously we don't want to be creating embryos willy-nilly. And so today we actually have genetically engineered humans. I'll make a note. We talked about CRISPR and we talked about human cloning. And in 2015, scientists decided that CRISPR on the human germline, which means your sperm and egg cells or in embryos, is something that shouldn't be done because we don't know what the effects will be. But doing techniques that are similar to cloning, you can actually treat you can treat diseases that exist in the mitochondria. So the egg that I showed you earlier has a nucleus, but it has the rest of its cytosol. So the nucleus contains most of the genetic material, but the cytosol has the mitochondria in it. The mitochondria have some of their own genes, and if those genes are bad, it can lead to metabolic disorders. So there's a technique that has been pioneered in Britain where you can have the mother, someone providing the donor egg, and you take the mother's nucleus and you put it into the donor egg, and you can, and now we have several children who have been born with that have actually been that have been born healthy, and they've avoided having defective mitochondria that would have severely limited their life. On the other end of the spectrum, in 2018, there's a Chinese scientist who wanted to make people make embryos who would be immune to HIV. And he was doing this to do this, he was taking a receptor out of their cells via CRISPR. Of course, this is kind of wildly unethical because these receptors, yes, HIV interacts with the receptor, but there's all sorts of other immune functions of this receptor that we don't know what would happen to. And so he was actually disgraced and fired from his job, and there was a major outcry from the scientific community that he would do this. Now, last year, I have a picture of this family where the baby in the middle was born with a congenital metabolic condition, and they were actually able to do a CRISPR-based treatment. And at least as of now, the baby is still healthy and not showing signs of the metabolic disorder. So this one is, I'll have to get back to you on that. I'm sorry. It's not diabetes. It's yeah, I don't remember the exact disorder in this case. I'm sorry. Anyhow. Now, this might, with the use of CRISPR in humans, you might ask, can we design babies with the characteristics that we want? And I'll note again, alert, this is wildly unethical because we don't often know the impacts of how messing around with our genes will carry through. There's also major scientific challenges here because determining the effect of the single gene on phenotype is actually quite difficult. In some specific cases, before the Human Genome Project, we thought, okay, if we knew what the genes were, we could know what they would do. And once we knew what the genes were, we thought, okay, we could figure out everything, we would have a roadmap. But it turns out that it's not so simple. A single gene, figuring out what it does, many genes in our body, we don't have a clear idea of what they do. And furthermore, I want to emphasize that phenotype is an emergent property arising from interactions between genes, as well as the genes themselves. So it's not enough to know, wouldn't even be enough to know the exact role of every single gene. And now I'll quote P.W. Anderson from his paper, More is Different, so some of the physicists might appreciate this paper. And he has a statement where he says, even if the elementary entities in science X obey the laws of science Y, that does not imply that X is just applied Y. So if we apply this to molecular biology, even if cell biology relies on molecular biology, cell biology can, because it is a larger scale science, is not simply applied molecular biology. And the same goes up to physiology of humans from cell biology. But this is something that motivates work at the center. I want to highlight Ben Kuznick-Speck, a former member at the center, Buduka, who you see every week, led this research to understand complex traits in humans based on their impact on gene expression. And of course, you see me and Professor Motter as well. Now, we've talked about cloning, we talked about individual things. I want to talk about de-extinction, and de-extinction has been in the news as of last year. This is the Hollywood Reporter from April 7th of 2025, and you can see the breathless title. Dire wolves have just been brought back from extinction, and no, this is not a story about Game of Thrones. But just over a month later, we get colossal scientist now admits they have not really made dire wolves. Now, the myth here was that these extinct animals were back again. The reality is a little bit more nuanced. They did do this isn't totally fraudulent, is what I want to emphasize. They did real work and made real progress because I mentioned earlier that it's hard to manipulate one gene, a lot of work to manipulate just one gene with CRISPR. They managed to put in 20 separate gene edits to make these wolves. And then they would have been born through a gray wolf surrogate. The problem, of course, is that they were finding these gene edits from ancient DNA and comparing it with the gray wolf. And it turned out that not every gene edit that you had gave rise to viable wolves. Some of them would be blind or deaf when they were born because you don't know what the functions of the genes are. Nevertheless, this shows the rapidly increasing ability to create, to direct evolution of species. So, next time you see something from Colossal, like the mammoth, where they have another project interested in exploring cold adaptation via the woolly mammoth and modern elephants, perhaps it's time to exercise some skepticism in the face of very slick marketing here. All right, so I mentioned chimeras before in the context of plants, and I want to get back to them. And for those who don't know, chimeras were an ancient Greek creature that had an amalgam of animal parts. And what I'm going to here, I am showing you a plant that is an amalgam of tomatoes in the scion and potatoes in the rootstock that you can see there. And this is actually something that is done commonly in agriculture, maybe not this specific example, but grafting roots and scions together is something that is done very commonly in, for example, in the apples you eat, because oftentimes if you allow apples to sexually reproduce, you end up with crab apples, not the delicious apples that you might have in your lunch later. So to do could we do the Greek chimera? Probably not. One, animals have non-self recognition in their immune systems. This is why it's hard to even do major problem with transplants between humans. And then furthermore, we have this problem of scale. Different animals are different sizes. Their organs are different sizes. And so the animal might fall apart because things would grow at different speeds. This is incidentally something that I understand is a potential problem in plants as well. Now, I want to show something that's work at the Center for Network Dynamics here. I think Joran is in the back. He did a lot of work on making this prediction. This is a chimera. It's on top, it has a tomato, as you can see, and on bottom it has chrysanthemum rootstock. Normally, chrysanthemums and tomatoes are incompatible. But if you put in tobacco interstock between them, you can now make these compatible. I believe Evan Eifler also contributed the actual experiments here, and you can see that this is growing happily since December, and we'll see how long, I don't remember how long we need to go for it to be considered a successful graft.
Oh, it has flowers now.
It has flowers now, so that's successful because it can reproduce.
Yeah. Okay. We'll wait to see whether it will produce tomato.
Okay.
And how much tobacco influence will be in it. Nicotine.
All right, are you going to send it for mass spec to detect the nicotine?
There'll be some.
Okay, so we have much to look forward to at the center. I also wanted to point out Nairi, in addition to Adelson, who spoke up, is obviously supervising this. Nairi Zerega is also in the center and working on this project. Now, can we do chimeras in humans? And I won't talk about all the possibilities of chimeras, but I will talk about xenotransplantation. This is something that's actually been around for a while, but there's a company that develops organs from pigs that have some of their genes changed to make them less likely to be rejected in humans. And as you know, many Americans are on dialysis, and if you've ever had the misfortune of being on dialysis, it's extremely time consuming and expensive. So if you could get an organ transplant, it would be a major change to your life. And so this gentleman, I believe it's Tim Anderson, received one of these genetically engineered kidneys last year, and he managed to live with it for almost 9 months. Interestingly, he was short of the record. The record is from 1964, and a woman named Edith Parker, who was a school teacher in Louisiana, received a kidney from a chimpanzee that lasted, I want to, a little over 9 months. Of course, we don't use chimpanzees anymore because of ethical concerns. So there's another gentleman who received his in September of last year, and I checked the news. There's no report that he has not, that it's been rejected, so we'll see if that one manages to break the record. Of course, this is I'll make a note on the ethical considerations. Some of this comes down to the values of how you view animals and whether that's a worthwhile thing to treat humans or not. Here, I think the ethical consideration is that you could help save someone's life, so you can take more risks than you would otherwise. But from now, I'm going to take a little bit of a right turn from talking about things that are based on molecular biology to talking about things that are kind of integrating our brains with machines. And I want to take you back to 2014. Some of you, especially those of you from Brazil, might remember watching the World Cup opening ceremony. You might remember the music, like we have Jennifer Lopez, Pitbull, and probably one of a famous Brazilian singer.
That's recognized.
Yeah, okay. No, I sorry. They also had a wonderful dancing and pageantry, but you might have missed this gentleman wearing a robot suit, and this is Julio Pinto, and he had had his spinal cord severed in a car accident 6 years earlier. And so he's walking in this robot suit to do the opening kickoff in the game. So how does this happen? How do they do this? Well, there's a group at Duke that has been researching brain machine interfaces over the last 20 years. And the idea is that you can take recordings from your brain and send them into a robot, and that robot can move. Brain recordings date back to the 1950s, so that idea isn't new, but trying to train someone to control the robot with their head is something that's new. And so you can have electrodes or an EEG cap that send signals that a computer can control. In the bottom here, you can see a gentleman with a robot arm who is picking up a glass. So here I'm going to show you some of the patients controlling a robot exoskeleton here. And you can see them, they're suspended. As with anything in robotics, you actually need about 5 people doing it. But you see Julio, kicking the soccer ball. And I think the thing I like about it is just how much joy the patients have in being able to move their bodies again, even if it's being done through the exoskeleton suits. Now, you might see these suits and think, can I get one of those? And in fact, you can't get one that'll connect to your brain, but you can get AI-enabled exoskeletons. This is marketing from a company called Hypershell, and caveat here, slick marketing from a company. Maybe don't believe everything you see, but I've heard from journalists who did demos at this year's Consumer Electronics Show in Las Vegas that these things actually work. They actually can reduce the amount of effort it takes to walk. Now, these are a little bit hard to see, but these braces that connect to their hips are the device. And so the idea is you have a battery pack, you have motors, and the device, excuse me, the device can actually sense the forces that you're putting on it. And so it uses machine learning to detect what you want to do and assist the motions you are trying to make. And then the motors that are in here help you with your motions. Now, can we push this toward can we push augmenting humans with machines in other ways? For example, can we do mind reading? And there's actually some research in monkeys that shows that you can actually predict the behavior of a monkey from the brain behavior. So this is from an experiment at University of Chicago where they took macaques and they put electrodes into the brains. And they could actually read the brain activity and predict when the monkey was going to move its arm to grab a device. And so is it in principle possible to do something like this in humans? I would say yes. Is it possible to read every thought that you have? I'd say the technology is definitely not there yet.
Which might be a good thing.
Which I'd say is probably a good thing. Which might be a good thing. But there are examples, if we can send signals to computers, can we send signals between brains? And in fact, this has been done in monkeys as well, where you have 3 monkeys and they're working to move a robot arm in 3-dimensional space. Each monkey is looking at a 2-dimensional slice of space, either the horizontal XY plane or one of the vertical planes, YZ or XZ. And the monkeys have to work together with their brains connected to the computer to move the robot. Now, there's not communication directly between the brains, but they can see the feedback based on what's on the screen to move the robot arm. And so the monkeys can actually work together and to make the robot move and recapture things. This has also been done with EEG caps. These are kind of caps that look like something you might wear in a shower or a swimming pool. They're non-invasive, but they have electrodes on them that can sense the waves that are in your brain. And so in this case, they had 3 humans playing a Tetris-like game. And so with their brain waves, they are steering these objects, and they're just like in Tetris, you're trying to make lines together. And so humans can be trained to synchronize their brain waves in this way as well. Now, again, a warning about marketing for companies. There's companies in the last decade that promise to upload your social media all to the cloud so that your children can have a digital avatar of you after you are gone. And you've probably heard of recent stories of ChatGPT trying to create personas of Albert Einstein or Isaac Asimov or other famous people from the past. It's probably possible to create something that feels quite convincing given how well large language models can emulate language. But speaking for myself, I don't know if it would be helpful for my children to have a digital avatar of me rather than to simply recognize that I'm dead. I'm not going to be physically present. So this is a little bit of a digression, but it's if you are going to exist forever, does that diminish the physical presence and put too much primacy on the emulation there?
I hope it can scare your children back.
I don't think they're paying too. Well, this is an Easter egg to see whether they're paying attention or not. So for one of the last ones here, I'm going to talk about. There's a lot of tropes that involve freezing monsters and then becoming unfrozen in a movie, like this scene from Godzilla. The voiceover for the movie might go, as global temperatures rise and the permafrost retreats, what horrors will be unearthed from the Arctic ice? So, with
Yeah.
is such a thing possible? Well, one answer is yes, because bacteria and viruses, if you remember from our first lecture, I covered this possibility that the Earth was completely encased in ice at certain ages of its history after life had arisen. So we believe that life probably has ways to survive bad times, potentially by laying dormant for thousands or millions of years. Oh, of course, we can't test the millions of years directly because we generally don't have access to living, like full living organisms from millions of years ago to see whether they can start growing again. But is this a real danger? Well, it can be. For example, in Siberia, there was permafrost that thawed, and there was anthrax in the soil. And this anthrax was the reindeer in the area normally would not be exposed to anthrax. They did not have the immune system capacity to deal with it. They got sick in 2000, died, and it even spread to one unfortunate human who also died. Now, this thing that's always scary about a million year old microbe wreaking havoc is that we have no natural immunity to it. And as people who lived through SARS-CoV-2, we know that not having any natural immunity can be a really big problem. On the other hand, unlike SARS-CoV-2, these would not have any exposure to us or our microbiota, and so it's pretty unlikely that they would be able to colonize and create pathologies in us. Now, another answer of yes is there's examples of organisms, and I talked about tardigrades, I believe, in the first lecture, that can enter static states to survive bad times. And we even have cases where in vitro fertilization, there's embryos that are left over, and there's some people who want to quote unquote adopt those embryos. And so as of last year, there was an embryo that was created in 1994 that was born to a healthy baby last year. So your embryos can live for a very, very long time. But there's a problem of scale here. Now, you guys probably are familiar with Walt Disney, and if you have learned that of this rumor that he was cryogenically frozen somehow. This apparently came out of a tabloid where a tabloid writer claimed to have gotten into the hospital and he said he saw Walt Disney suspended in some kind of metal tube. Unfortunately, while this would make a great story, it's not true. There's no evidence that Disney was frozen. But this is a common, we see there's lots of pseudoscience on being like frozen at the moment of death until they come up with a treatment for your disease. Could you survive? And it's probably not, because we don't know how to restart brain activity after it stops. And brain activity can't go on after your heart stops when you're still alive and not brain dead. But we also know from people who undergo, cardiac arrest, within minutes after of not getting enough oxygen to your brain, it starts to die and your brain damage becomes irreversible. So there's this whole part of keeping the brain ready and reanimating it that we know nothing about. And then the second issue, this is more physical, is that freezing and thawing would probably result in tissue damage. And to see this, you can look at all of the construction that will happen in the next few months. Because construction season is almost upon us as it gets warmer, because the asphalt expands and contracts with the temperature, leading to cracks that need to be repaired. A similar thing would happen with the expanding and contraction of tissues, because we're mostly water, and when water crystallizes, it would create issues. But maybe we don't want to, let's walk this back from like fully reanimating people. Could we like pause life, kind of like maybe the bacteria do? Because, for example, if there's a disaster like a nuclear war, maybe we'd want to be able to have people enter a state of stasis so that they could come back after the effects of the radiation had worn off. Or maybe we were traveling between the stars and we don't want people to have to be alive for the trip through interstellar space. Well, hibernation is something that we know that many large mammals do. And we've also seen that there's also treatments where we introduce hypothermia or comas in people with traumatic brain injuries to limit the amount of damage. So theoretically, hibernation should be possible in humans, but we don't know much about how animals induce and maintain hibernation. So currently, probably off the table. Another problem that we know from hibernation is that when animals wake up from hibernation, they're not well rested. They're actually sleep deprived, meaning that there's normally during sleep, we are cleaning damage in our brains. So potentially staying asleep or staying in hibernation or torpor for a very long time could potentially be damaging to the brain as well.